Do you know specifically about people that maybe stop taking the course? No. Okay. Okay. So, first of all, how many did someone didn't get a chance to read the article or look at it at all? Did you? Anyone? So what did you think about the article in general? First of all, it's an old article. So that one of the things I want to emphasize is that if you I don't know how many articles you've already hey, I don't know how many articles you already read, but the language was a little bit different than what we're used to today, and I mean that it's a lot more straightforward, and a lot of times it's much easier to read the articles that are from these times because they really write what they found and they write the logic of what they're doing. In contrast to today, when a lot of time it needs to be more suggestive. Everything is like, everything you write has to be like a little bit more subtle. You can't state obvious things, and then it's.
After the first page, it really did become easier because we practically wrote down everything. But even though we did start learning about G proteins and stuff like that, I felt that the first page and a half were still a little difficult, and that was the hardest part.
Yeah, it was actually the first, because it's again it's experiments that you are not familiar with, and that's also what I will emphasize more on, the electrophysiology and stuff like that. I will focus a little bit less on because I think you'll discuss it in detail in other courses and throughout your academic career also. So let's start with the guiding questions or general questions that are important to understand this article. First, before we start, Eric Kandel, which is one of the writers on this article, is one of the, like I told you, the forefathers of neurobiology, also a Nobel Prize laureate, and his work during the years was very, very, very systematic. He studied this specific pathway of activation. Specifically, he focused on the serotonin, the aplesia, and the effects up to the memory, up to the changes in the synapse, and to the level of the memory. And it's really beautiful to see how throughout his academic career he really followed this very, very clear path, focusing on one pathway and really understanding it thoroughly, which is also very different from what a lot of times we do today. So, first of all, what is CRE? Or we'll start from what is CRE, for the second question. And I gave you a hint here. Yeah?
It kind of goes together. CREB is a transcription factor that binds to CRE, which is a certain DNA sequence, and therefore, through CREB, you can manipulate the transcription of the protein after that sequence.
Right. So, like you said correctly, it's a lot of time the reason I wrote these hints is that a lot of times the things that you don't understand are the most basic questions and basic information, and also a lot of time that's the hardest part to find out. For example, if you look in articles, if you're trying to find in articles what is CRE and what is CREB, it will take you especially if it's like a basic concept, then it will take you ages. You will see the writing CRE element, like, and nobody will tell you CRE is a DNA segment or DNA sequence that CRE knows how to bind to. So that's why Wikipedia is so great, obviously, because it's really necessary to get this base level. So, like when we say promoter, when we talk about promoters in general, then it's important to understand that a promoter is a sequence. It's a sequence in a gene, or part of the regulatory sequence of a gene. Okay, so am I assuming correctly that you are familiar with the term LTP in general, more or less? So long-term potentiation. A lot of time people, and also we'll talk about this in relation to this article. Long-term potentiation is basically a mechanism that was defined or was established in very simple neuronal systems, like in the hippocampus, in which you have a very clear input and output method to detect. Like you have okay, we'll talk about it later, but in general, the potentiation is the ability of the synapse to change and thus strengthen. Meaning that if I gave a stimulus, I got a specific some degree of response, and after the potentiation, for the same type of stimulus, I will get a stronger response of the postsynaptic cell. And specifically, what is the ELTP and LLTP? Yeah? Mm-hmm. Right. So E is early, L is late. So it's late phase long-term potentiation and early phase long-term potentiation, which specifically in this article is one of the first examples where the researchers here showed that there's a difference between these two features. Okay, and that what was considered like LTP in general is not a very exact way to describe the term LTP, and there are other processes that may look the same at the beginning but differ in their late phase. Okay, so in what way did the RAB subunit differ, or was it different from the wild type unit of the PK? Yeah.
Half from the article, half from Wikipedia.
Mm-hmm.
Is it true that ELTP doesn't require protein synthesis and LTP does?
For the time of this article, it's true. Okay, like today, I will discuss. I will talk later a little bit about what from here is still relevant today, which is a lot. But there are a lot of concepts that in the past they didn't even consider. The researchers didn't even consider. For example, one of the things that you see all the time is the immediate coupling between transcription and translation. Okay, they always say like they interchange the terms. They say transcription and translation is required, and they show. And actually, in the article, they only show that they inhibited the protein synthesis. They didn't show that they inhibited transcription of RNA, but they refer to it as it's the same thing. Okay, that's part of the approach that really drastically changed in the last 10 years, for example, which is not looking, and we'll learn about it in the next chapter, that the central dogma in what we call DNA, RNA, protein is not just a continuous sequence, that the RNA in the middle also matters, and matters a lot. Okay, so it's not and one of the reasons that is that a lot of time RNA can be much like a lot of time after it's been transcribed, it can be silenced or kept in the silent form and just be accessible for ribosomes for protein synthesis later or in different parts of the cell. So that's one of the. In certain types, in the hippocampus, in this certain example, the early phase of LTP, which they describe here, has a very strong component that doesn't rely on protein synthesis. Okay, in the early phase, it doesn't mean that there's no protein synthesis or there's no function for the protein synthesis. For example, I don't want to get to, but we're going to talk about it in the next chapter. But keep that in mind for now. So again, to this question, specifically one of the terms that I also added in the slide I added for you to understand better the article was the term negative dominance. So did you understand what this means, like in general? Like dominant, negative. Like one of the things that they say in the article is that this subunit, this mutated subunit of the pKa, was negative dominant. And what it means, and this is a very we didn't get into it at all, but this is very, very basic genetics. You probably heard about recessive traits and dominant traits, right? That you have certain traits that are dominant, certain traits that are recessive or not dominant. And it's actually a very, very complex term, and it has it's a lot more complicated than what you normally learn in grade school biology. But in general, it means that if you have a certain type of gene, a lot of times it will overtake or overpower the function of the second, because we have two copies, right? We have two copies of each gene in general. Then the dominant form means that if it's present, it means that the activity will look as if only that form is active. Okay, meaning that if I have the best example is from plants, and if I have if I have a certain flower that here has the gene that produces red color, and here I have a gene that produces white color, then if these two are different, the only difference between these two are specific mutations. Okay, so let's say you have mutations here that cause red color instead of white color. Okay, so these are considered different alleles or different copies of the same gene. Let's say this is let's invent the name of the gene, CLR4 gene. Okay, this is the color gene that determines the color of the flower, and it has two variants. One of them is the red variant, and the other one is the white variant. And if a certain individual or a certain plant has these two white copies, he has two copies of the white gene or the white allele, then the flowers will be white. If it has two copies of the red, then the flowers will be red. And this is only determined experimentally a lot of times. If it has one copy of this and one copy of this, if the flowers are red, completely red, like the same level of red as they were in this condition, then we would say that this allele or this copy of the gene is dominant over the other. Okay, so it's a dominant form over the other. And there's also all the states in the middle. Okay, it can be a lot of times you see conditions where if you have these two copies, then the flowers will be pink, for example. Okay, so it's very complicated. Okay, in every type of every specific example is very, very different from one another. There are many mechanisms that determines dominance over the other. Sometimes it's one form really actively silences the other form, and sometimes it's just that you need a certain. It can be mechanisms that are like that, and even when we talk about epigenetics, it's not. Maybe I'll add, if we'll have time at the end of the year, I think I'll add a part about epigenetics, because I see it's coming up a lot, but it's not actually, this is not what we refer to as epigenetics, okay? But I'm just giving an example of certain mechanisms. No, so again, I don't want to get into that, okay? But for example, you can think about it. Let's say you have a different enzyme that needs like the activity, the determinant for whether the flower will be red. It just needs like a level of the protein over a certain amount, okay? And let's say it just needs a level of if it like this produces 50 units of red of the okay, and this produces 0 units of red. Okay, and you have somewhere down the line you have a sensor that if you have more than 30 units of red, then the output is red. Okay, for example, and if you have so it's enough to have one copy of this in order to cross this threshold or this molecular threshold, and you get a red phenotype or a red expression of this form, and this is one of the mechanisms. But actually, I think that in this case, they don't know what is the exact mechanism. They think that in the assembly of the pKa, because it has these two subunits, then one of them assembles a lot more efficiently, and thus it comes into or is expressed a lot more than the second form. So, and also you need to pay attention that a lot of time the wild type form or the natural form is a lot of times not the dominant form. Okay, so it's a little bit hard to imagine that, but this is like very a crash course on hereditary genetics or Mendelian genetics in general. So generally what it means here is that when we say that the RAB subunit, which is the mutated subunit of the pKa, when we say that it's negative dominant, it means that if it's expressed in the cell, then most of the activity, or most of the, if we now check the pKa molecules that we have in the cell, most of them will have this subunit. Okay, so this subunit will be expressed, and it will be in all the proteins or the pKa in that cell. Yeah.
A natural form that just like once every I don't know thousands of pKa there's this form and they just took it and made sure that it was inserted into a lot of forms, or is it a system that they cause themselves?
Actually, I think I don't know specifically. That's the first answer. I don't know specifically for this one. I went a few articles back to when they started this to when they started using this form of this mutated form, and they didn't get to that place where they described how they discovered it. But in the article for sure, and even in the previous article that they used the same variant, one of the researchers here, they didn't describe the method at which they produced it. Obviously, if you have, they told that one of the mutations here is in the nucleotide binding site for the cyclic Aamp, and they could theoretically, if they had the structure of this protein, they could theoretically also, like we saw in the example of the NMDA receptor, they could target a specific mutation to a specific location. Although the methods that we have today to induce a single mutations are much more powerful than what they had 20 years ago. Okay, so it's a lot easier today. Okay, what did they use the CAMK2 alpha promoter or CAMK2A promoter here? Okay, so but what is the reason? Like, what is the logic behind that? You're right. First of all, you're right in what you said. But what is the logic behind using this?
To control where cells the RAB will be expressed.
Right, because one of the things that you want to do, especially if you want to study a very specific system for, and in this example, it's the hippocampus circle or hippocampus circuitry. What you don't want to do is affect the whole animal, Okay, and this is one of the first examples, actually, in the literature that they use this approach. And today it's very, very, very common. Okay, it's very common to use to piggyback ride on a gene, on the CAMK2A gene here, that you know that has a specific expression pattern. And the specific expression pattern here is that it's specific to the brain, first of all, and it's specific to frontal regions and to hippocampal regions. Okay? For example, where doesn't it express? In this article, in the amygdala, right? So it actually does express in the amygdala, but in much lower levels. Okay? So the logic behind this, and it's pretty amazing actually that this thing works, says that, where's the eraser? The logic behind it basically means if we have a gene, and this is the CAMK2A, okay, and this is the CAMK2A gene, it means, okay, we saw that this protein is expressed in the regions of the brain that we want to now affect, then probably somewhere here, somewhere upstream, and if we're looking at this is the direction of the gene, then this is considered to be upstream. And again, if we're talking about strands of DNA, and this is the 5' and this is the 3', this is also always the directionality of the DNA. Then, and I'm not showing here the signal. Okay, I'll show the reverse strand. So this strand is 3' and this is 5', just to be exact. So what they assume is that somewhere along here there is a promoter sequence that specific transcription factors that are only expressed in the cells that we want to express our gene know how to bind specifically to. Okay? So what they did actually here is that they didn't know what the exact promoter is. Nowadays we know a lot more about specific promoters. So they actually took a huge chunk of the segment of the DNA that is upstream of the CAMK2A gene. They took like 8,500 base pairs, which is a lot larger than what you need to take today.
How many are usually promoters?
Promoter regions are usually 1,500 to 500. Actually, promoter regions sometimes could be also inside the gene. Okay, not necessarily you have to have upstream. Okay, so it's like the normal size, the typical size is about 2,000 base pairs, which is a region that specifically in here you have specific sequences that transcription factors like the CREB, but it's a different transcription factor that binds here, knows how to bind to and express this gene. So what they actually did is that they took this whole segment upstream of the gene, and now they can input it and put their gene. They can put their gene after it and cause specific expression only in these regions of that gene. The amount of gene expressed is it is in general. The promoter also determines the amount of that transcription. Okay, but there is also many other factors that determine it. Okay.
I wasn't sure I understood this, but mice with specific knockouts didn't show these symptoms, and they thought that it was because there was overcompensation. So I thought maybe they need to.
They talked about, that's one of the things that I talked about, which is different writing from what you see today. Along this article, they referred a lot of times to like a different model that was created where they did a different manipulation to the pKa, and over there they didn't see a behavioral phenotype and they didn't see any effect. So a lot of times they refer to that research of a different research group in order to say their research was not good, their research was not, the reason that we see an effect and they don't see an effect is because they did this wrong. Okay? That's what they say in general. And they don't say that specifically, but from their language you can understand that, but even this language is not accepted today. Okay? The language that they're using in this article, you will not find it today. Normally, like a researcher will try to find excuses for why maybe his model fits also models of other groups and try to be very, very polite in that sense. And here they were not very polite. Okay, so what is the role of cyclic AMP in this pathway? And this is not directly from the article, but yeah? Okay, so the cyclic AMP in this pathway is a secondary messenger, like we saw in the previous class, meaning that if you have some kind of signal that activates the G protein coupled receptor, then in turn the G protein can activate, and we'll learn about that in the following class, in the next class, about how exactly this process happens, in which it starts from a specific receptor, then goes through a G protein, and this G protein activates an enzyme that can change the amount of this cyclic AMP, and that's why we call it the secondary messenger. And this cyclic AMP is actually what binds to the PKA, and what releases the PKA from its inhibition and renders it active. Last question is, what type of signal do we know that can trigger LLTP? From this article, for example.
LLTP is triggered by activation of Aamp, which can be produced in several ways, like several neurotransmitters, like specific G proteins that are activated by Aamp. They give the example of dopamine, but there are more. And also calcium. It's kind of like that.
But also what starts the signal, and they showed it in the article, is like these tetanic pulses, right? They did this tetanus pulses, these 4 pulses that also activated this signal. So now we're gonna, so after we went through these questions, I think it will be easier for you to understand this article, so we'll look at it a little bit in more detail and try to understand the rationale of this research and what's new about it. Okay, so for example, a lot of times nowadays it's very common to hear people change the terms LTP with memory, like interchange the words, like LTP is memory. Okay, but this link is really not obvious. Okay, in general, like LTP is some physiological phenomena that happens at the level of the neurons, and memory is like something very abstract and not really necessarily related to these two. So one of the major things that this article did, and this what also makes it iconic, is showing for the first time one of the most in the beautiful examples of the link between LTP and memory in general, and showing these two processes. They are two together, and also they defined in a very exact way, or until this paper it wasn't known exactly how. But the role of the PKA in this long-term memory, or this long-term memory consolidation. So, also one of the things that I like about this article, that's the logic behind it, I know for me, is very clear. What they did is very simple. They created like a model, and in the end, and after they created this model, they just passed it through a series of tests to see what are the phenotypes that they see from here. So the model that they created is transgenic animals that have specific reduction of the activity of the PKA only in the hippocampus. And like I said before, they did this with the special construct that they built. Because of this specific reduction of activity, they saw that the LLTP is decreased in the CA1 region in the hippocampus. And also behaviorally, they saw that animals exhibited normal short-term memory, and but these animals experience long-term deficits or memory deficits, and specifically in tasks that are required for what are considered like hippocampal tasks. Okay, and they say hippocampal task because this is another one of the things that today we understand a little bit more. Like in the past, people say that memory is hippocampus. Okay, but today we know that memory is not only hippocampus, and the hippocampus has a specific role, especially in memory consolidation. But for example, much of our memory, or if I can, and a lot of it is also unknown, much of our memory is stored in the cortex, for example. Okay? And also in the cerebellum and other areas. So, and the last thing that they showed here is that the time course, or the time it took to ruin this ability, or to the time course parallel, like they said here, looked similar to wild-type animals that they were treated with protein synthesis inhibitors, meaning that this whole process is probably mediated through protein synthesis. Okay? And specifically, I mean here protein synthesis because they didn't show the test for transcription. Question? No. Okay. So what do we see here? Do you know? What are these? How are these experiments called in general? These. Okay? It's important when you look in the first question that you need to ask yourself when you're looking at the figure is what is measured here? Okay? What exactly am I seeing here? Okay? And first of all, this is a mouse brain, obviously, and it's cut like in a mid-sagittal section, not mid-sagittal, but in a sagittal section, where you can clearly see the hippocampus. And these are two different genetic lines, two mouse lines that they used, and they differ from each other actually in the amount of the copies that this gene or this genetic element was inserted in the genome of these mice. Okay, so actually, and this is also one of the things that really improved during the last 20 years through the better implementation of these genetic tools, is to control how many copies you will get of this gene that you knocked in or you inserted into the animal's genome. Like in the method that they used here, they couldn't really control the amount of transcript that they inserted into the mouse genome. So they actually, like, you can say, like, stuffed the mouse genome with here 7 copies of this transcript and here 16 copies of this transcript. But what they claim is that the activity change was still very specific. Like nowadays, you will not see an article that has such a gross or such a non-sensitive way in order to knock in an animal. You can really control that and also insert, like in a specific location, this transcript, and also insert just one of these transgenic elements. Yeah, probably what they. A lot of times, the location of in the DNA really changes the expression. For example, like we saw in the previous examples, that you have areas of DNA that is more condensed and less condensed, like aerochromatin and heterochromatin. And if you knock the gene into a place where the DNA is packed, then it wouldn't matter that you have like a specific promoter or et cetera, and because this area of the gene of the genome in the specific cell will be closed, and it won't, and no transcription factor can get to it. Okay, so it's actually important that what we call the genetic environment of where you insert your transcript is very important for the expression profile. Okay, so probably a lot of these 7 inserts here and a lot of these 16 inserts here are probably not expressed at all, and just several of them which fell into open areas got expressed. Probably they checked many mouse lines, much more than they described here, and just in specific ones. Yeah, they said 3. Okay, but probably there were like 10. Okay, but and they just picked the ones that showed the effect that they wanted. Like, okay, so how exactly does this what does this tell us? And this is like a very general introduction to genetic techniques that we'll discuss in the end of the semester. But if you see something like this is a very simple representation. Then here they describe they put the CAMK2A promoter, again a huge region, and this arrow signifies like a place where transcription starts from. This is a transcription start site, a general example of that. Here they have a special intron that they put inside. Do you know why they put this intron here? So actually there are certain... What? An intron is a segment of DNA that during the process of the initial mRNA processing, it's getting cut out or cleaved out or spliced out, like they want to say, of the mature mRNA. And in the end, the mRNA that's getting shipped out of the nucleus doesn't have this element inside it. Okay? Then, actually, again, what I'm trying to show you here, and that's why I'm also focusing on this construct over here, is that in order to express certain genes, the story is not that simple. It's not that you put a specific promoter, you put your gene, and you have expression. A lot of time you have these elements that you need to add in order for the system that you introduce this artificial transcript, in order for it to really express this transcript. For example, one of the things that nowadays people don't use this method of this intron anymore, but in the past, this intron over here is actually what helped stabilize this transcript and made sure that it started. What? It's just trial. In general, it's just trial and error. It's observation. It's what was accepted in that time. If you want to do, like, again, it's hard to say when this all started, right? Because this is based on like research of 50 years before. So I don't know where in this process people started looking at and saying, hey, if we put this intron before our gene, then it gets expressed better. Okay, so I don't know exactly when this happened, but this is like one of the things that roll around the scientific community, and that the information that's passed through the scientific community that people are reporting. Listen, if you put this intron over here, then your transcript here will be more stable. And this is also one of the reasons that they think, I can tell you that molecularly, we think that a gene that gets alternatively spliced or gets spliced is being kept is because it has to go through the whole series of the processes that normal mRNA go through. Okay? So if most mRNAs in humans, 95% of all genes go through splicing. Okay, they have these introns inside them and they go through splicing. So the logic behind it means that even if you don't need to have splicing and you put just the construct that you want to get expressed, a lot of time if you wanted to fit into the normal secretory or the normal processing pathway of the mRNA, then you need to add this element here, so it will introduce splicing, and your transcript will start will like fit into the process or the natural pathway of the cell. Okay, so it's like yeah, in mammalian in general. For example, that's one of the major differences between us and yeast. Okay, so we have a lot of genes that are similar between us and yeast. But actually, yeast has very low level of splicing and alternative splicing. Okay, so, and I didn't talk about what is cDNA, but basically cDNA is just. Well, we talked a lot about this. Okay, so maybe I'll go forward or ignore the part about cDNA. I'll just talk about here the polydenylation is again. In order for, we didn't talk about it, but we'll talk about it in the end of the semester. But in order for the mRNA to be stable, it has to have a polydenylation tail or a poly-A tail, meaning that when you have an mRNA, it has to have poly-A's that are added to the 3' end of the molecule, and this is one of the mechanisms that help this mRNA from degradation. Okay? Because, for example, and this is just to show maybe part of the logic, a lot of the viral genes don't have polyadenylation, and also don't have certain elements in their RNA, and a lot of the nucleases or the proteins that degrade nucleic acid work from this side of the molecule. Okay, so if you have a longer poly-A tail, it means that this molecule will be stable longer. Okay, so one of the things that they added here is a polyadenylation signal. They actually didn't add the poly-A by themselves, but they added a signal that they know induces long polyadenylation in the cell. Okay, so all these elements together, this element and this element, are here to stabilize the transcript here, and this element over here is to cause this specific expression that we see here. In this example, we see an experiment of in situ hybridization, in which what they actually stain for is RNA. Okay, they want to see where do they have expression of the genes of this transcript. So they designed a specific probe or specific molecule that they can further identify, and I'm not going to get into that too much. That is specific to this segment or to the RNA that is produced by this transcript. And what they saw is that indeed it had very high expression around the hippocampal regions and frontal regions of the mouse brain. Okay, this is the hippocampus of the mouse. And in the wild type, they didn't see anything. Okay, questions? So this is important for the proof of concept that they actually did what they claimed they did. Later, they estimated the PKA activity specifically in the hippocampus, in a specific region of the hippocampus. And here you have I just plotted here the model of the PKA the mutated PKA that they have here. So it has a different subunit that's mutated in 2 regions, and thus leading to lower activity of this PKA. So in the absence of cyclic Aamp, which is the activator of this protein, the levels in the mutants, in both mutants, were lower, okay, than the one of the wild type. And also when they added cyclic Aamp, then there was a significant reduction in the levels of these in these 2 transgenic mice relative to the wild type.
What is
PKI, okay, it's good that you asked. PKI is a specific protein kinase inhibitor. Okay, it's a specific molecule that they know that inhibits specifically protein kinase A, and that's important here in order to show that the activity that you see here is actually PKA activity and not some other kinase activity. Okay. For example, the way they do this assay, actually they did it in a very different way from what people do today, but in general, it's based on a concept in which you take your substrate, like for example, you have a protein that you know that PKA phosphorylates, you add a kinase, and then what you add is, in their case, they added radioactive labeled ATP molecules, meaning that the third phosphate of the ATP was radioactively labeled, that you can later detect. And actually what this kinase does is transfers the phosphate group from ATP to the substrate. So you have this phosphorylated substrate that has a marked radioactive phosphate group linked to it. Then you can wash the sample from all the kinase and the free ATP, and then you can detect the level of how much phosphorylation you had on your substrate that was not washed away. Okay, there are different ways to do this assay. You can do it with a gel. You can do this by binding to a plate. You can do this in many different ways, but I don't want to get into it too much. But this is actually the method in which, where you measure the activity, like how many proteins get phosphorylated in general. That's what you ask. So here's a general introduction of what they tested next. They looked at after they saw a reduction in the activity of pKa, they looked at the early phase LTP and late phase LTP, which basically in this experimental paradigm means if you do 1 or 2 trains, then you get early LTP, and if you do 4 trains, you do late LTP. Okay, so it looks a little bit funny, right? Like if you do what happens if you do 8 stimulations, right? What happens if you do 20 stimulations? So again, you need to understand like the context, and in this context, what they see, what's very different between these two processes is that this one decays very rapidly, and this one can stay up to 24 hours, or or as long as you can keep your slice, like they did here. So this potentiation, or the change in the APSP, remains for a very, very long and stable time in the late phase. So I'll just discuss this too, and then we'll take a break. What is the point? What is the point here that they wanted to show here in these figures in general? Okay, so fibril amplitude is again is a method to estimate to estimate like the change in the response, if they measure the slope in the FEPSP, is the change in the response? Well, they measured it. I don't want to get into that too much, but it's 2 different measurements. Let's say it's just 2 different measurements, but we don't have a lot of time, so I don't want to get into that too much. Again, I said I will not focus on the electrophysiology, so you have to forgive me about that. But in general, what they saw from here is that they didn't see any change in general between the wild type or the mutant mice. Okay? That's what they want to show here. What? Yeah. They measured that even if they do 1 train of this tetanus pulse, like we see here, which just means like repeated pulses. Okay, we repeated the high frequency or medium frequency pulses. If they have 1 train of tetanus pulse or 2 trains of tetanus pulse, there is no change in the potentiation, in the level of potentiation and the level of the decay of the potentiation between the wild type and the 2 mutant mice. Okay? Where did they see a change? In the late phase. Okay? So the long term or the late phase LTP, after 4 trains of tetanus pulses, they saw that that also initially, but mainly they focus on this late period. They see that in the mutant mice, the signal or the strengthening of the connection between the 2 regions that they checked decayed, and in the wild type it didn't decay, and it stayed constant up to the point that they could measure from the slice. Okay, and what is this here for? Do you know? A lot of time when you see these traces next to a slide, it means that these are like controls, or these are like typical representations. So this is a typical. They want to show you these 2 traces are from somewhere around here of all the animals. Like the lower one is a typical EPSP. Actually, I think no. I think this is a typical EPSP before training. Okay, for these ones, and this is a typical EPSP after 180 minutes. Okay, so you can see that after the decay, the EPSP looks pretty much the same for the 2 mutants, and for the wild type, it looks drastically different. Okay, the response that they measured from the wild type is drastically different after this period of time. Okay, so we'll take a break, and then we'll continue discussing what they found. Okay. So we're running a little bit late. Like it took a little bit more time than I planned, but it's nevertheless it's good. Like the questions that you're asking about these are very critical for this course. So don't hold back. Okay. Like if you have questions that are regarding to like genetic things that you don't understand and stuff like that, this is exactly the aim of this course. So specifically ask these questions. Okay, so like I said here, the main objective here is to show that there is no effect in the transgenic mice. In the transgenic mice in the short-term LTP or the short-term processes of LTP. But they did see a change in the late phase or LLTP. And later on, what they claim is that this change is due to the effect of the pKa that later causes, that is mediated by protein synthesis. Okay, and they'll show it in the end. But what is also unique and what's nice about this article is that, like I told you before, they walked the full path, like we want to say, like from the from the molecular mechanism that they studied or they tried to do, they went all the way to the behavioral test, and even more than the behavioral test. Probably, many of you have studied like psychology, they even differentiated between two types of memory, and and actually. Probably what happened in this article in the order that this research was actually done is that, like I told you, they probably had like many multiple lines of animals, okay, and just the ones that showed the effect behaviorally. Later on, they went on and tested the effects molecularly, okay. So a lot of time the order that you see in articles is actually the opposite order that the actual logic in the experiment went through. So, are you familiar with the Morse water maze? Like you read a little bit about it now, but did you hear about it before? Okay, so the Morse water maze is one of the most common methods to evaluate like spatial memory in mice or rodents in general. And I'm not going to go over this method. Again, I put you a link in Wikipedia, which I hope is sufficient. But what they actually found in these animals is that that these animals learned much less, like they had impaired spatial memory relative to the wild-type animals, meaning that they were almost the same amount of time in all the different quadrants of the Morse water maze, or the pool of the Morse water maze, compared to the wild type that were that spent most of their time near the target quadrant, near the target quadrant where the platform was in their training period. Okay, but what they actually found is that in foot shock experiment, in contrast to that, they didn't see a change. In foot shock experiment, they did two types of experiment. One of them was context dependent, and I'm not going to get into like the specific how they did these two experiments because I don't want to get held back on that. But in experiments that are context dependent or experiments that are cue dependent, then they saw specifically that there was effect only in context dependent after 24 hours, meaning that the immediate context learning was not affected, but they saw a dramatic difference in the context dependent learning 24 hours after, meaning that the mutant mice, like forgot, okay, the effect of this foot shock or this fear conditioning or that was induced to them before. And by contrast to this task, in the more, like you say, more classical conditioning task, which is associated to areas of the amygdala and not the hippocampus, here they didn't see any difference between these animals. So you can say like it's not a general impairment in memory, which is a lot of time it's very hard it's very easy to mess up something in biology, okay? A lot of time you mess up one specific factor and you have a whole, like it can, if you had like this deficiency in general memory, then we could always say like, okay, maybe they just screwed up the metabolism of the cell. For example, something that's not related to memory at all. If the cell doesn't have a sufficient amount of energy, then again, new connections are harder to form, for example. Then this could be like an alternative example. But here, it's very, very important to see this example in which they have only specific types of functions that were impaired, and these functions are more related to the hippocampus. Like this function is related to the hippocampus, and this function is more related to the amygdala areas. And last, and more, this is last, yeah. And importantly, what they showed is that they can achieve similar effects if they take wild-type animals, and that's the reason that you don't have here the RAB mutants. You just have wild-type animals that either were induced with saline, which is the substance that you dissolve this molecule in, or they had a saline together with this molecule that inhibits protein synthesis. So if they administered this molecule half an hour before the context-dependent task, then they saw that 1 hour after they didn't have any difference in the learning, but 24 hours after they had a massive impairment in the learning, and here it was not specific to the area. Okay? So what they claim is that protein synthesis is important for long-term facilitation. It doesn't matter in which system, in the amygdala or the hippocampus, but the specific function that they ruined here is specific to the hippocampus. That the context-dependent long-term memory is facilitated through the activation, like a large activation of PKA. Okay, and when they ruined this ability, then they impaired only the specific function in the specific region. Okay, and the specific type of memory, like you would say. So, like in any great or in any good article, you always have a model in the end that's supposed to like summarize everything, and even things that didn't appear in this article at all. For example, here they show the short-term potentiation mechanism that are facilitated through CAMK2A, for example, which they didn't talk about in this article at all. But what they divide here is that they say, okay, there are two types of memory that we know of. There's short-term and there's long-term, and the immediate long-term, or what they say immediate long-term, is facilitated probably through a mechanism that the PKA effects can have, like, or similar molecules can have directly on channels and similar processes. Like, for example, they say that there could be other protein kinases that may be involved in direct phosphorylation of certain channel, and thus facilitate like this early long-term potentiation. But in contrast to that, in order to facilitate long-term and really long-term potentiation or late phase LTP, you need to have transcription of new genes or production of new genes and new mRNAs and new proteins. Okay, and again, remember that what they showed here is protein, but they regard to it like it's the same process. Okay, when they're talking about transcription, they're talking about protein synthesis. Okay, and again, is there something here that bothers you in general in this model? I'll give you a hint. Does a neuron look like that? No. What's different? No, something big, okay? Something like, how does a neuron, if you draw a neuron, how do you draw it? Do you draw it like that? You have a neuron and you have synapses, okay, you have synapses on the neuron, like this. Okay, you have axons and dendrites, okay? Why don't they draw it here?
They don't know if this happens in the cell or the axon.
Okay, so first of all, one of the problems here is that if they claim that you need, because one of the things that they don't talk about, or the elephant here in the room, which today we have a solution for this elephant, but in the past they didn't address this issue, is that these are very, very much far away from this area. Okay? Meaning, like in certain types of cells, for example, like in mortar cells, these are basically detached from one another. Okay? Like the distance from here to here can be 1 meter. So one of the things that they don't want to discuss, that they don't want to deal with, is how is this effect that has to go through the nucleus, how does this affect, or the genes that here get to the synapse? Okay, and this is one of the things that they don't talk about, and that's why it's comfortable for them to show a model that the cell looks like something. Everything is very close. You have this pathway for short, you have this pathway for long. Okay? But we'll discuss like this is one of the drastic things that we know today much more, the process that we know much more about than what they knew in the past. Okay, so this took much more time than I thought, but never mind. Again, if you have questions regarding this article, feel free to ask. Specifically not on the electrophysiology, if you have about the genetic techniques and everything that they use here. This is actually one of what's nice about this article is that they really use a lot of techniques that are still used today. Okay, today we know how to do them a lot more exact and more controlled way. But the yeah. Elaborate about that. We're going to talk about that in the end of the semester. Okay? So, okay, let's very, very briefly, or maybe not, I don't know. Let's very briefly go over the questions that regard the last chapter that you should have discussed about. One of the problems first of all, I want to apologize about the language of these questions. I think I wrote it in very late, and I was also sick or something like that. Because later when I looked at these questions, I realized that some of them didn't make a lot of sense. But still, let's go over them very, very rapidly. Below are 3 steps from operation mechanism. Of a protein that transfers iron from one side of the plasma membrane to the other. So you need to have these 3 components, which means ion binding, conformational change, and the release of the ion. And which type of these transport proteins falls into these 3 conditions? Does this fall into this condition? Voltage independent ion channel? No. Why? Well, that's a question. Because it does bind ions, okay? But, for example, it doesn't undergo conformational change, okay? No, the voltage independent doesn't undergo conformational change, and this one of the reasons that this question is not very good, okay? Like, what is exactly ion binding? What is conformational change? Okay, so never mind. ATP-driven pumps need to go all 3 of them. Okay, what about symporters and antiporters? Okay, so all of them undergo all 3. And voltage-dependent ion channels? Again, it depends what you refer to as ion binding, because they do undergo conformational change that makes them open, and they do bind and release ions in general. Okay, so it depends which type of channel, because we learned about specifically certain types of channels that bind specifically the ions, and there are channels that are just permeable to the ion with the water surrounding it, so it's not exactly ion binding. So it's another very great question in general. In what aspects do different ion channels differ between them? What's different between different ion channels? In the type of the transferred ion? Yeah, in the transfer rate? Yes. Dependency and dependency on the membrane potential? Yes. And dependency and dependency of a ligand? Yes, great. Okay, during the transfer of potassium ions across, and I should have said the resting K channel, the channel that we learned about, The ion is surrounded by water molecules? Yes or no?
Yes. No.
During the transfer?
No.
No. Okay. And I know we talked about that. You said you told me that you learned about different models and stuff like that. Okay. So I checked it and I looked everywhere, and all the models that are referring to this channel, some of them just differ in like that. You have also a water molecule, a single water molecule that passes through a channel while the other ion is passing, but it doesn't matter. In general, all the models are talking about that the ion is dehydrated, meaning that it loses the water molecules. Okay, in this specific channel. In the voltage gate channel, for example, it's not in this. It doesn't go through this mechanism. So the ion is shed the water molecule envelope, and is there something else here that's right? The ion interacts with other ions in the channel. Well, is the oxygen an ion? So the oxygen is not charged in general. I should have wrote in here interact with other components or other molecules inside the channel, but never mind. Okay. So in the operational mechanism of voltage-dependent sodium channels, the refractory period is achieved due to? Right. Okay. So the conformational change is not what causes the refractory period. Okay. What?
But it does. I mean, the global region is a conformational change.
But not by the movement of the potential sensor. Okay. Yeah, it moves, but in the beginning when it moves, it doesn't necessarily, like, these two movements are not correlated in a very well. Like they're correlated in time, but they're not affecting each other. It's actually like the voltage in general. Like if you have the probability, it's another mechanism. Yeah, you could say that it's another potential sensor in general. Okay, like we learned, if you hyperpolarize the membrane, then you have more release of these globular regions of the channel. But again, that's not a very great question, like you stated. Okay, last question, which is okay. Which of the following is a mechanism for termination of synaptic signal? Is A, diffusion? Yeah, but not a very efficient one, and normally not what happens. B, yeah. C, not really. Maybe, but we don't know. And D, hydrolysis of a neurotransmitter, in which example did we see this? The D? Right. Acetylcholine, which is hydrolyzed by acetylcholine esterase.
It can terminate this signal.
Yeah, you're right. It's true, but like I would probably accept it. But yeah, I'm aware of that. I told you that the questions were written like bad, but maybe I should have said like which of the following mechanism for termination, for immediate termination of synaptic signal. Okay, so diffusion outside of the synaptic cleft is much slower. Okay, for example, than the reuptake. For one of the reasons that you don't really appreciate, and maybe we should focus on this a little bit, about how the synaptic cleft actually looks like molecularly. So how it actually looks is that you normally draw it like this. Okay? So when you look at it like this, then you say, okay, so a neurotransmitter can go here, or it can go here, right? So it's diffusion. How the neurotransmitter gets from here to here? It's strictly diffusion. Okay? So if you look at these models that this is how they draw synapses, then you can believe that diffusion is maybe a relevant force here or relevant process here. But actually, how a synapse really looks like is, and I'll draw it with my special technique. okay, is more or less something like this. And here you can understand why. And here you can understand why the diffusion, if a molecule is here, then the amount of time it will take for it to diffuse is very long relative to the amount of time it will take it to cross the synaptic cleft. Okay? And even here, I'm not exaggerating enough. This can be much more much more complex. And I'm not even regarding, do you remember what I showed you about the vesicle that's covered with protein? So the same thing is about the synapse. So the synapse has these many proteins that are spanning these two regions and create this mesh or this forest of protein in general that also makes it problematic for diffusion in the lateral or in the horizontal direction. And also, specifically, there's a huge difference between this ratio between humans and mice. This is actually one of the things that people don't know, is that in humans it can get to up to 10 times more, the surface of touching between two synaptic clefts, for example, just because the neurons are a lot of time much larger, and also the contact area that they have between the two synapses are really different. Okay. Mm-hmm.
And like a lot of neurotransmitters are amino acids.
Yeah. Well, in general, phosphorylation needs to. It's not that it sees amino acids floating around; it phosphorylates it. It has to have normally the context of the full protein. Okay, and the protein in its configuration, it has a specific like. So. Again, we get into the debate in biology if we talk about the general rule or the exceptional. Okay, the term that I'm not aware of mechanisms of phosphorylation of neurotransmitter in the synaptic cleft, and it doesn't mean that there are no mechanisms that can phosphorylate neurotransmitter in the synaptic cleft, and also I don't see like a mechanism in which. But maybe there is, but in general we need to be very clear, especially in this course where we're talking about the general phenomena. The general phenomena is just by these two mechanisms. And even this mechanism is pretty rare and is specific to acetylcholine. So this mechanism is like the main mechanism at which the termination of the synaptic signal occurs. Okay? Yeah. Yeah. Yeah, maybe like the syn-porters and anti-porters also undergo conformational changes. Okay, that's what you're. They can also bind. Like it refers to the question of what do you refer as binding. Okay, you have like specifically, a lot of times in the syn-porters and anti-porters, the actual binding is what causes the conformational change. So the binding is like very specific and also specific to a certain type of ion. Okay, and they can bind multiple ions also, like we saw in the example of the anti-porters, that they use the binding of one ion to transfer a second ion and vice versa. Okay, so here specifically there is very strictly saying specific ion binding. Here, like I told you, like the ion binding is, you still have ion binding, but you don't have like this conformational change that is happening due to the ion binding. Okay? Okay, so now we'll have to go very fast. And last class, we finished, the last slide we saw is this one, right? Okay, so this I'm not going to talk about. So, no. Okay, so what is a G protein in general? again, in the signaling molecules that we discussed, or the signaling overview that we discussed, one of these pathways of the signaling, one of the signaling pathways is mediated by G proteins, okay, or G protein-coupled receptors, and specifically a G protein is normally a trimeric protein, meaning that it has 3 subunits: the alpha subunit and the beta gamma subunits. Normally they're referred to as together because the beta gamma bind to each other in a very tight manner, and they also operate together. Which, in contrast to the alpha subunit and the beta gamma subunit, that during the operational mechanism of the G protein, they detach from one another. Okay, so all G proteins also have, normally in the alpha subunits, they have what we call a GTPase, or a GTPase region, which means that they hydrolyze or GTP to GDP. And these membranes, although they're not shown here, are anchored to the lipid bilayer. Okay, they have this anchor over here. You see it in the model shortly, that anchors them to the membrane. So they move by diffusion along the plasma, the inner plasma membrane. Okay, so how do G proteins actually function? In the resting state, when you don't have any hormone or ligand that is bound to the G protein coupled receptor that we can see over here, then that trimeric G protein is assembled and is associated together. And also normally you have a GDP molecule or what is in D-phosphate molecule that is bound to the alpha subunit. Okay, so in this state, everything is resting, nothing is active. What happens after the hormone binds to the G protein coupled receptor is that the receptor undergoes a conformational change. This conformational change causes the active receptor to associate with the G protein, with the assembled G protein, and this association alters the ability to the G alpha subunit to bind GTP. Okay, instead of the GDP, now it binds a molecule of GTP, guanosine 3-phosphate, and the binding of the GTP induces a second conformational change now inside the G protein itself, which causes a dissociation between the G beta alpha subunit and the G alpha subunits. Okay? When we have a G alpha subunit that is bound to GTP, we regard to it as an active G alpha protein, which means, or G alpha switch, and I will say why we refer to it as a switch, which can then diffuse across the membrane and bind to specific proteins that it now activates. Like this binding specifically activates effector, what we call in general effector proteins. Okay, so in the we'll show specific examples of this mechanism. And again, this process can repeat itself again and again and again, as long as you have active receptors, or you have a hormone or ligand that's bound to the G protein coupled receptor. So, for example, you could have like the G alpha activate this effector, then detach, and then move back. If the receptor is still active, move back, bind another GTP, and vice versa. So what you actually get here is and I'll show you an example of that later, is that you get because the GTP initially, like, it doesn't stay long in the form of GTP. It gets hydrolyzed and it switches back to GDP. And when you have the GDP, then the state in which these two proteins associate is favorable, and you have the reassociation or reassembly of the G alpha to the G beta gamma. So it's not available, and also it doesn't have any potential energy in order to activate the active effector. So I think we don't have time for this specific movie, and we'll just switch to the next slide. In general, there are two classes of GTPase switch protein. And I will show you in the next slide why exactly we call it switch proteins. And we have the trimeric G protein, which we saw in the previous example, like this one, that's composed of 3 subunits. And there are also monomeric G proteins, normally from a family that's called RAS proteins. And they look generally like the alpha subunit, but they're not, they don't associate directly with the G protein coupled receptor, but they're linked indirectly by certain adapter proteins. Okay? And these two are very distinct pathways in G protein coupled receptors, and they're controlled and regulated in very different ways. But in general, I think we're just going to focus on this type of proteins, which are the trimeric G proteins. So why do we call? Yeah, the difference is that in this case, the G protein is composed of 3 subunits. In this case, the G protein is just 1 subunit. Okay, that's homologous or is similar to the alpha subunit of the trimeric G protein. Okay, so it's not just that and these are 2 distinct families of G proteins. They have distinct function and they're also regulated differently. Okay, but we're going to focus just on the trimeric G proteins. So this is an example or molecular example of an alpha subunit of a G protein, and the reason that we call it a switch is that, like a switch in the light, okay, if we want this room to be illuminated, okay, then if we flip the switch, the energy from flipping the switch actually doesn't give the energy for the light, right? The energy of the light comes from a different source, but we just triggered a conformational change, right, in the circuit that rendered this circuit as active. Okay, so these G proteins, these are actually what they do. They don't strictly transfer energy for the reaction itself, but they transfer energy, but they hold energy in the form of this potential energy between these 2 switch subunits, or not subunits, switch domains of this protein when they're bound to GTP. And this energy, or this potential energy, is what causes a conformational change in the target protein, or in the effector protein that they activate. Sometimes not activate, but sometimes in the opposite way. But this triggers the downstream effect in the next protein in line that alters the activity, the general activity of that protein. Okay, so when it's bound to GTP, it's not active, it doesn't have any potential energy, but when it's bound to GTP, it has this potential energy through the binding of these side groups to the phosphate, to the 3rd phosphate here. So, in general, in most cases, the effector protein is activated by the G alpha GTP, or the G alpha bound to GTP, but in some cases it could be inhibited also. Okay, so this conformational change doesn't necessarily mean that the effector protein will be activated. It could also be inhibited. Okay, and in rare cases, the actual effector, or the subunit of the G protein that affects the effector protein is the G beta gamma subunit, and not the G alpha subunit. Okay, like we'll see an example shortly. Very common pathways or common systems in our body that use G protein coupled receptors are, for example, odorant receptors in the nose and rhodopsin receptors in light, which we will also focus on. And you can say that one of the logics behind it, especially in the olfactory receptors or the odorant receptors, is again but maybe we'll talk about it. You know what? We'll talk about it later. In general, when we talk about the odorant receptors in the nose, each type of odorant activates a specific type of G protein coupled receptor. Okay? So, for example, if we're sensitive to 300 types of smells, then we have 300 types of different genes that code for 300 different types of G protein coupled receptors, that each one of them is sensitive to a different type of odorant. Okay? For example, the mouse has like 1,500 genes, and we have like 300 genes. Okay, so we have much less genes that code to odorants than the mouse. And obviously, the full sensation of smell is achieved by a combination of many different types of odorants and the activation of many different types of receptors. Okay? But what's important about the system in general is that it's very much combinatoric, but still animals that are more reliant on smell, like mice, have much more receptors and much more genes that are responsible to sensing different types of odorants. Okay, so the combinatorics in their case is much larger than in our case. Another thing that the system enables, and we'll see an example of that shortly, is that it enables a very drastic amplification of the signal. Meaning that if you want to detect, for example, as we'll see in the example, a single photon, and if you want to have a response for a single odorant that you recepted, then if you strictly just open one ligand gate in ion channel, for example, then the effect will be really small. But because of the G protein coupled receptors, then you can have a drastic amplification of a signal of a single photon in this case, or a single molecule in the case of the odorant receptors in the nose. And how exactly does this? Okay, so this, I'll skip this. I'll show you this in the end. So, a specific example of G protein coupled receptors is the case of cyclic Aamp, like you saw in the article, and specifically the epinephrine example. So epinephrine, or generally in its popular name, adrenaline, has different effects on different organs in the body, and the effects are mediated by different types of receptors that this ligand binds to, or norepinephrine binds to, in different organs of the body. For example, in the if this epinephrine molecule binds to beta-adrenergic receptors, it causes catabolism of glucose and massive production of ATP. It increases, again, the heart concentration contraction rate, again, through beta-adrenergic receptors. But if it binds to alpha-adrenergic receptors, in contrast, it can cut off circulation of unneeded organs, if we're talking about fight or flight response, in the stomach or kidney, for example. Like if you want to escape a bear, then you don't need your stomach and kidney at this specific moment. Okay? So these are two opposing effects, and they actually have two opposing effects also molecularly. Like different types of receptors have two opposite effects when we look at the molecular level. Actually, beta receptors, what they do, activation of these beta receptors over here, if they bind to a epinephrine molecule and ignore the fact that these molecules look different, they're the same. So if this receptor binds to an epinephrine molecule, what it does is activates a G protein coupled receptor, a specific type of G protein coupled receptor that activates at the NAC class, and thus causing an elevation in the levels of cyclic Aamp. Okay? And again, what I mean here when I talk about molecular switches is that while this protein is bound, while the G alpha subunit is bound to the NAC class, the NAC class keeps producing more cyclic Aamp molecules. Okay? And the energy for the production of the cyclic Aamp doesn't come from the G protein. Okay? So it really acts as a switch, like you think about it. If it's bound to the NAC class, then it produces cyclic Aamp all the time, and if it's not bound to the NAC class, then it doesn't produce cyclic Aamp. An opposite effect is through the alpha-1 adrenergic receptor, in which a different type of G protein, if it binds to the same NAC class, it can cause an inhibition of the activity of the NAC class. Okay, which means that normally you have this basal level of activity of the NAC class, and if this G protein binds, or G alpha, in this case it's G inhibitory alpha, binds to the NAC class, it causes an inhibition in the production of cyclic Aamp. Okay, in the rate of the production of cyclic Aamp. So these two phenomena, or these two receptors, contradict each other, and normally what we see also cellularly, in most responses to ligands, you can see both responses in the same cell. A lot of time you have, and what determines the net effect of the response is the abundancy that you have of these receptors relative to these receptors. Okay? So in biology in general, normally you have all the components of the reaction, and the net effect that you see in the end arises from the abundancy or the effect of each reaction separately. Okay? So here we have a movie that will summarize this, and I think I'm not going to finish with that.
Imagine you are hiking through the woods on a late summer's eve when you have the misfortune of coming across a rather disgruntled grizzly bear. Given the scarcity of food in these parts, he is delighted to have come across such a palatable entree. A series of transient depolarizations in your neural circuitry identify the bear as a predator and quickly decide that running away is your best option. In order to elude your pursuer, you'll have to react quickly at both the synaptic and cellular level. To accomplish this, a number of signaling molecules are released to various targets throughout the body. When faced with this type of stressor, the brain sends a series of messages that lead to activation of neurons in the peripheral nervous system. This leads to release of a neurotransmitter called norepinephrine. But how does this chemical signal get translated to a cellular response that will allow you to escape? The effect of the chemical signal depends largely on the type of receptor to which it binds. Ionotropic receptors are located in the cell membrane and have a pore that allows ions to pass through. Once inside the cell, the ions can alter the local membrane potential and increase or decrease the likelihood of an action potential. A different class of receptors is the G-protein coupled receptor, or GPCR, which are part of a group known as Metabotropic Receptors. As the name implies, these are paired with a G protein, or guanine nucleotide binding protein, which is composed of 3 functional units, an alpha subunit and a beta-gamma dimer. Normally, the G protein is bound with a guanosine diphosphate, GDP molecule, and is inactive. Unlike ionotropic receptors, the GPCRs have no pore. Rather, their efforts are mediated by activation or inhibition of other molecules. When a signaling molecule, for example a neurotransmitter, stimulates the GPCR, the receptor essentially changes its shape, allowing the G protein to bind to its intracellular domain. The alpha subunit then exchanges its GDP for a guanosine triphosphate, GTP, creating an active state. This results in the G protein subunits disassociating from the receptor, as well as each other. The alpha subunit then binds to an effector protein, such as an enzyme, which then acts to increase or decrease the production of a second messenger. These so-called second messenger pathways can lead to a number of possible changes inside the cell. This is perhaps the main difference between ionotropic and metabotropic receptors. While the effects of the GPCR are typically slower than those of the ionotropic receptors, they are much longer lasting. An astounding number of second messenger pathways are found in our cells. One example is the cyclic Aamp-dependent pathway, which, as in our example with our hungry bear, may be activated by the neurotransmitter norepinephrine. In this case, norepinephrine binds to a specific kind of GPCR called a beta-adrenergic receptor. Once this receptor is stimulated and the G protein activated, the alpha subunit binds to an enzyme known as adenylyl cyclase. This in turn increases the production of cyclic Aamp, which is the second messenger. Cyclic Aamp will go on to activate protein kinase A, pKa, which in turn regulates a number of different proteins. Ultimately, activation of the cyclic Aamp pathway may influence processes such as cell survival, the up or down regulation of gene transcription, and the production of several neurotransmitters and receptors. Now, back to the bear. At the moment that you first encountered it, your flight or fight instincts decided that it was in your best interest to run. The activation of diverse pathways in the brain ultimately results in a surge of norepinephrine, which binds to the beta-adrenergic receptor located in many of your organs. This causes your pupils to dilate, your heart rate goes up, and blood circulation is increased to your muscles to push them to their limits. It does this through activation of intracellular signaling pathways, such as the cyclic Aamp pathway we have just seen. So, by binding to this specific GPCR, norepinephrine allows you to escape, despite the odds.
Okay, so apart from the apparent problems in this video, again, one of the things that I want to show you in general this course is the difference between like popular science movies in general and how you see normally proteins and how you see and how it actually happens. For example, I don't know if you saw, but in the end it showed like the pKa molecule and the proteins that it phosphorylates, like the small ions that are floating around. So this is not the case at all. And again, one of the things that they don't show in this movie is the negative or the adverse reaction of the inhibition through a different type of receptor. So you always have to think about that the response is much more elaborate than what normally you see, and you have many types of like feedback loops that regulate the regulation on top of the initial response. So we'll just finish like this specific example, and then we finish for today. So we discussed that the norepinephrine or the epinephrine that binds to the specific receptor activates and elevates the amount of cyclic Aamp. Cyclic Aamp, like you also saw in the article that we talked about, activates pKa, and this is actually the active part, or the kinase part of this enzyme. In this part, it just causes dissociation between these two factors, and the release of this factor is actually what renders it active and available to phosphorylate other proteins. In general, when I talk about amplification, then in G protein coupled receptors, specifically in the example here of epinephrine, but it's also true in many other cases, you have 4 levels of amplification of this response. So, for example, one epinephrine molecule can produce many can activate many adenylyl classes, because I told you that during the time that the ligand is bound to the G protein coupled receptor, the G protein coupled receptor can activate many G proteins. Okay, and these G proteins, in turn, after activating many adenylyl class molecules, each adenylyl class molecule can produce many molecules of cyclic Aamp. Okay, again, the example that I gave you for the light. Okay, it's not that the G protein gives the energy for this reaction, but it just turns this reaction on. So in this step, you need 2 cyclic Aamp molecules to operate to activate one protein kinase A, and 2 other levels of amplification that we have here is that each protein kinase A, once it's activated, it can phosphorylate many target proteins, and these target proteins, once they're phosphorylated, like I told you in previous classes, phosphorylation a lot of time are also like molecular switches. Okay, phosphorylation also renders many enzymes and many proteins active, and thus these proteins can then downstream affect many processes down the line. Okay, so you see that the binding of the epinephrine in this case causes this cascade of responses that are amplified at least 4 times during this very simple scheme. So just one last point is that how this reaction or how this process is getting terminated. maybe I'll talk about it next time. Okay, so in general, the termination of this response happens through, we can say, three main processes or three main factors. One of which is that the affinity of the receptor hormone to the hormone decreases when the GDP bound to G protein is replaced by GTP. So this conformational change not only affects the G protein, but it also affects the receptor itself and causes it less, like the affinity or the binding capacity of the receptor is decreased. Moreover, cyclic AMP phosphodiesterase, which is an enzyme that breaks apart the cyclic AMP, starts operating very, very fast because the level of cyclic AMP are elevated. So this protein can now degrade the cyclic AMP in the cell and turn it to a non-active linear form of AMP. And prolonged activation can lead, because one of the things that I told you is that in the end you have pKa that phosphorylates many types of enzymes, and one of these enzymes can then phosphorylate, or sometimes we refer to it as autophosphorylation, but this is not the case here, that it can phosphorylate the receptor, and the phosphorylation of the actual G protein coupled receptor renders it unable to bind to the G protein coupled receptor, to the G protein itself. And this is actually shown here. So if you have a G protein coupled receptor that is phosphorylated, so initially, if you have like partial phosphorylation, then it interferes with the binding of the G protein that's supposed to bind in this domain, domain of the protein. And if you have sufficient phosphorylation, in this example, you just need to have two sites of phosphorylation. Then you have a third type of protein that's called arrestin, which you can infer from its name, that it just bounds in a very specific way to this region over here after it was phosphorylated, and this completely stops the ability, or completely blocks the ability of the G protein coupled receptor to bind to new G proteins. Okay, and just a minute. And also, in cases that the activation is too severe and you have massive amount of phosphorylation, it can also cause endocytosis of this whole region inside the cells and actually like deplete or reduce the amount of receptors that you have on the membrane. Okay, so if you have like a membrane that has this receptor over here, then, and you have the arrestin molecule that's bound over here, it can actually cause a process of endocytosis, like we saw in the recycling of the synaptic vesicles, and this will now be engulfed inside a vesicle that will be endocytosed or internalized inside the cell, and thus lowering the amount of receptors in general.
This is like a specific mechanism? Is it similar for other hormones?
This is a very common mechanism. The epinephrine molecule is just an example here, but we'll see in next class the example of that in the visual system, for example, where you don't have a hormone at all, you just have a photon that activates the whole system. But these are the main pathways that cause this reaction to stop. Okay, so next class we're going to finish this chapter and we're going to start the next chapter. And we'll talk about G protein coupled receptors that regulate ion channels, and in the end, the activation of gene transcription by G protein coupled receptors, like we saw in the article.